Have you ever wondered how identical twins, right, with the exact same DNA, can sometimes end up with, well, different health issues or even just look or act differently as they get older?
Yeah, or think about this. You get one copy of a gene from your mom, one from your dad. Usually both work,
Hmm.
but sometimes only one copy is actually switched on, while the other one stays completely silent.
It's fascinating, isn't it? It kind of challenges the simple view of genetics we sometimes have. Okay, so let's really dig into this today. We're taking a deep dive into something that helps explain these puzzles, this amazing field called epigenetics.
Hmm.
Our mission really is to explore this hidden layer of control. What is epigenetics? How does it actually work on a molecular level? And crucially, what impact does it have on development, disease, even how things we experience might affect our genes, maybe even our kids' genes?
And what's so cool here, I think, is that epigenetics gives us a way to understand how traits or gene activity patterns can be inherited without changing the actual DNA letters, the sequence itself.
Right. It's all about gene expression, how the genes are used, not just what they are.
Exactly. So think of it like this. You've got your genome. That's your fixed blueprint, right? Your DNA sequence.
Mm-hmm.
But on top of that, you can have many different epigenomes. These are like different sets of instructions or markings that tell your cells which genes to read and when. And these epigenomes can change throughout your life. Different cells have different ones.
And they change based on what?
The environment.
Yeah, environmental cues, development signals,
Yeah.
all sorts of things. And the really mind-blowing part is that some of these epigenetic marks can actually be passed down, which is why it's so vital for understanding development, diseases like cancer or diabetes, even, behavior and how organisms adapt.
Okay, so how does it work? Like chemically, what's happening? You mentioned three main ways.
So, broadly speaking, there are three core mechanisms. First, there's a reversible modification of DNA itself, usually adding or removing a little chemical tags.
Okay, tags on the DNA. Second, chromatin remodeling. That's about changing how the DNA is packaged up with proteins.
Making it easier or harder to read, I guess.
Precisely. And third, there's regulation by non-coding RNA molecules, RNA that doesn't make proteins but still controls genes.
All right, let's tackle that first one. DNA modification. You said methylation, like adding sticky notes.
That's a great analogy, yeah, DNA methylation. And the complete set of these methyl sticky notes across your whole genome at any given time is called the methylome.
The methylome, got it, and it changes.
Constantly. It's different in your brain cells versus your skin cells, for example. And it adapts. When a cell gets a signal, its methylome can change in response.
So how does the cell actually add these notes?
Well, in mammals, there's a family of enzymes, DNA methyltransferases, or DNMTs. After DNA copies itself, or as cells specialize, these DNMTs add a methyl group that's the tag onto a specific DNA base, cytosine.
Creating something called 5-methylcytosine, 5-MC.
Exactly. The little 5-LC makes up maybe 1.5% of all our DNA bases. It's a key epigenetic signal.
And where do these tags usually land? You mentioned specific spots.
They have a preference, yes. They mostly go on cytosine bases that are immediately followed by a guanine base. We call that a CpG denucleotide.
C next to G, CpG.
Right. And often you find lots of these CpGs clustered together in areas called CpG islands. And importantly, these islands are frequently located near the start of a gene in the promoter region.
Ah, the promoter, the part that kind of turns the gene on or off.
Exactly. So what does the methylation do there? Well, if a CpG island near a promoter is unmethylated, the gene is usually active, ready to be transcribed.
Okay, open for business.
But if that CpG island gets methylated, if it gets those sticky notes added, the gene is usually silenced, turned off.
How? How does the tag silence it?
It works in a couple of ways. The methyl groups themselves can physically block the proteins needed to start transcription from binding to the DNA, or they can attract other proteins that lock down the DNA structure, making it inaccessible.
So it's a pretty clear mechanism for switching genes off.
It really is. And while promoter methylation is key for silencing specific genes, it's worth noting that most methylation in the genome isn't actually at promoters. It's in repetitive DNA sequences, kind of the junk DNA, especially in tightly packed regions called heterochromatin.
And why methylate that stuff?
That helps to keep things stable. It silences potentially disruptive elements, like bits of DNA that can copy and paste themselves around the genome transposable elements. Methylating them keeps them quiet, which is vital for preventing chromosome problems.
Right, okay. Is 5-MeLC the only game in town for DNA methylation?
Good question. For a long time, we thought so, especially in mammals. But recent work in other organisms, like some algae and worms, found they might not use 5-melacy much, but instead they methylate a different DNA base, adenine, creating 6-mela.
6-mela. And does that happen in mammals too?
People are actively looking into that now. Whether 6-melody exists in mammals and what it might be doing epigenetically is a really hot area of research. Still unfolding.
Fascinating. Okay, let's move to the second mechanism. Chromatin remodeling. You said this is about how DNA is packaged.
Exactly. Our DNA is incredibly long, right? To fit inside our tiny cell nuclei, it has to be packaged very efficiently. It's wound around proteins called histones.
Like bees on a string?
Kinda, yeah. A segment of DNA wrapped around a core of 8 histone proteins forms a nucleosome. And then these nucleosomes coil up and up, forming chromatin, which eventually makes up our chromosomes.
And when it's all tightly packed, the genes are hard to access.
That's right. The default state for many genes when packed into dense chromatin is off. So chromatin remodeling and histone modification work together to control accessibility. They can loosen the packaging to turn genes on or tighten it to turn them off.
How do they do that? Modify the histones?
Yes, the histones themselves have these flexible tails that stick out from the nucleosome core. These tails are key targets. They can be chemically modified in over 20 different ways.
Wow. Who does the modifying?
Specialized enzymes, we call them writers. They add the chemical marks. Erasers remove them. And readers are proteins that recognize specific marks and then trigger downstream effects, like recruiting other proteins.
Can you give an example of a modification?
Sure. A really important one is histone acetylation. Adding acetyl groups, little chemical tags, to specific spots on the histone tails tends to loosen the chromatin structure.
So acetylation generally means on. Gene activation.
Generally, yes. It creates a more open chromatin state, making the DNA accessible for transcription. And conversely, removing those acetyl groups, deacetylation tightens things back up, leading to gene silencing. It's a dynamic balance.
I've heard about something called the histone code. Is that related to all these modifications?
It absolutely is. The histone code is this idea that the combination of different modifications on the histone tails acts like a complex signaling language.
Like different marks mean different things.
Exactly. It's not just if a histone is modified, but which specific amino acid on the tail is modified, how it's modified, acetylated, methylated, phosphorylated, etc., and what other modifications are nearby.
So a specific pattern, like say H3K27E3, that means something specific.
Precisely. H3K27E3, that's lysine 27, on histone H3 being trimethylated, is often a strong signal for gene silencing. It recruits reader proteins that help maintain that silent state. Other patterns signal for activation. It's incredibly complex and sophisticated.
Wow. Okay, so we have DNA tags and histone modifications. What was a third mechanism? Non-coding RNAs.
Right. Non-coding RNAs, or ncRNAs, these are RNA molecules made from DNA templates, just like messenger RNA, mRNA, but with a crucial difference. They don't get translated into proteins.
But they still do important jobs.
Oh, absolutely. They play many roles, and a big one is in epigenetic regulation. They can help guide enzymes to modify histones or DNA. They can help structure chromatin, and they can directly silence genes.
What kinds are there?
We can group them by size. There are short ncRNAs, less than about 30 nucleotides long. These include things like microRNAs, miRNAs, and short interfering RNAs, siRNAs. They often work by targeting specific mRNAs for destruction or blocking their translation, effectively silencing the gene after it's been transcribed.
And there are others.
Yes. Also, Pee-wee interacting RNAs, pRNAs, which are really important in germ cells, sperm, and egg for silencing those jumping genes, the transposons we mentioned earlier.
Okay, those are the short ones. What about long ones?
Then you have the long non-coding RNAs, or LNCRNAs. These are much bigger, over 200 nucleotides. Structurally, they can look a bit like mRNAs, maybe get processed similarly, but again, no protein product. And we've found thousands of LNCRNA genes in humans.
So what's their epigenetic role?
LNCRNAs are often key epigenetic initiators or scaffolds. They can act like guides. Imagine an LNCRNA binding to a specific spot on a chromosome and also binding to a histone-modifying enzyme.
Ah, so it brings the enzyme to the right address.
Exactly. It targets the epigenetic machinery to specific genomic locations. By doing this, LNCRNAs can orchestrate changes in chromatin structure over quite large regions, leading to coordinated changes in gene expression patterns. It's a very precise level of control.
Okay, so we've got these three mechanisms. DNA methylation, histone modification, non-coding RNAs, all working to control gene expression without changing the DNA sequence itself. Let's talk about where we see this in action. What about cancer? How does epigenetics fit into our understanding of cancer?
It's a huge area, and our understanding has shifted dramatically. For decades, cancer was viewed mostly as a disease driven by accumulating genetic mutations, changes in the DNA sequence.
Right, mutations in genes controlling cell growth.
Exactly. But now, there's overwhelming evidence that epigenetic changes are just as critical, involved in both starting cancer and helping it progress.
Okay, how does DNA methylation, those sticky notes, factor into cancer?
It's complex because methylation patterns go haywire in cancer in two main ways. First, there's often global hypomethylation.
Meaning less methylation overall?
Yes. Compared to normal cells from the same tissue, cancer cells often have significantly lower levels of DNA methylation across the genome.
And what does that do?
It can cause chaos. It can lead to the inappropriate activation of genes that shouldn't be on, including genes that promote growth or cancer development. It can mess up the silencing of imprinted genes. And crucially, hypomethylation in those repetitive DNA regions can lead to chromosome instability rearrangements, changes in chromosome number, which are classic hallmarks of cancer cells.
So less methylation is generally bad in cancer, but you said two ways. What's the other?
The flip side is hypermethylation, but it's targeted. While the overall level might be lower, specific regions, particularly CPG islands in the promoters of certain genes, become more methylated than they should be.
So targeted silencing.
Exactly. And guess which genes often get silenced this way?
Tumor suppressors.
Precisely. Genes that normally put the brakes on cell growth, or repair DNA damage, or trigger cell death when things go wrong. Genes like BRCA1 in breast and ovarian cancer or MLH1 in colon cancer. Their promoters get hypermethylated, silencing the gene. This is a major way cancer cells disable our natural defenses.
So the cancer cell loses its brakes epigenetically.
Right. And these hypermethylation patterns can be very specific to certain cancer types, sometimes even helping doctors diagnose the cancer or predict how it might behave. It shows how these epimutations, these abnormal epigenetic marks, can be just as important as DNA mutations in driving cancer.
So is it fair to say cancer is often a mix, a combination of genetic mutations and these epigenetic screw-ups?
That's exactly how we view it now. Cancer arises from an accumulation of both genetic and epigenetic alterations. You might have one copy of a tumor suppressor gene knocked out by a DNA mutation, and the other, perfectly normal copy, silenced by promoter hypermethylation.
The end result is the same. The protective function is lost.
Right. Both hits disable the gene, allowing uncontrolled cell division. We see this interplay constantly in cancer.
And presumably, histone modifications and chromatin remodeling go wrong too.
Absolutely. The writers, erasers, and readers of histone marks are often dysregulated in cancer. Their abnormal activity leads to messed up histone profiles that contribute to the cancer state.
Okay, here's a really hopeful question then. If epigenetic changes, unlike DNA mutations, are potentially reversible, does that offer new strategies for treatment?
That is the really exciting part. The reversibility of epigenetic marks is a huge focus of drug development. It led to the whole field of epigenetic therapy.
The goal being.
The goal is to develop drugs that can correct these abnormal epigenetic patterns in cancer cells, to demethylate those silenced tumor suppressor genes, or to readjust the histone modifications, essentially trying to reprogram the cancer cells back towards a more normal state.
Are there drugs like this already?
Yes, several have been approved, and many more are in clinical trials. For instance, drugs like azacitidine, also called Vidaza, are used for certain types of leukemia. It's basically a fake cytosine that gets incorporated into DNA and traps the DNMT enzymes, leading to less methylation.
So it helps turn those silenced good genes back on.
That's the idea. And then there are drugs targeting histone modifications, like histone deacetylase, HDAC inhibitors, such as vorinostat or Zelenza. They block the enzymes that remove acetyl groups, hoping to push the chromatin towards a more open active state for tumor suppressor genes.
How effective are these epigenetic therapies?
They're showing promise, definitely. Right now, they're often moderately effective on their own and frequently used in combination with traditional chemotherapy or targeted therapies. It's still relatively early days.
So what's the future direction?
A lot of research is focused on understanding why some cancers respond better than others, finding the best drug combinations, and identifying epigenetic biomarkers, specific marks that could predict who will benefit from these therapies or who might become resistant. It's a really dynamic and hopeful area of cancer research.
It really is. Okay, shifting gears slightly, but still on this theme of change. Epigenetics isn't just about disease, right? It also connects to how our environment shapes us, maybe even future generations.
Yes, absolutely. This is perhaps one of the most profound implications of epigenetics. It provides a mechanism for how environmental factors, things like diet, stress, chemical exposures, even social interactions, can leave a lasting mark on our gene expression patterns by altering the epigenome.
Can you give a human example?
A famous, though tragic, one comes from the Dutch famine near the end of World War II. Women who were pregnant during that period of severe starvation had children, the F1 generation, who, decades later, showed significantly higher rates of certain health problems: obesity, heart disease, even some cancers and mental health issues.
Compared to people born before or after the famine.
Exactly. And what's more, some studies suggest that certain effects, like altered growth patterns or increased obesity risk, might have even persisted into the next generation, the F2 grandchildren.
Wow. That suggests the environmental impact was somehow transmitted.
It strongly suggests a transgenerational epigenetic effect, though the exact mechanisms in humans are still being worked out. Animal studies provide clearer evidence.
But which ones?
There's a classic set of experiments using mice with a particular gene called GUDI. A mutation, called AV, involves a piece of mobile DNA jumping in near the agouti promoter. This usually leads to mice with yellow fur, who also tend to be obese and prone to diabetes.
Okay. And the epigenetics.
Here's the cool part. The researchers found that the level of methylation on that inserted piece of DNA varied dramatically, even between genetically identical AV mice.
So same gene, but different methylation levels.
Right, and this directly correlated with coat color. Low methylation meant yellow fur and obesity. High methylation resulted in mice with mottled or even completely normal brown pseudo-agouti fur, and they were healthier.
Identical genes, different outcomes, driven by methylation variation.
Exactly. These are called metastable epialleles alleles, where the epigenetic state can vary and be inherited, influencing the outcome. And here's the kicker. They fed pregnant yellow AV mothers a diet supplemented with nutrients known to support methylation, things like folic acid, vitamin B12.
And what happened to the pups?
The offspring were much more likely to have the brown pseudo-agouti coat color. The diet shifted the epigenetic state towards higher methylation, reducing the prevalence of yellow, unhealthy mice.
That's incredible. Diet directly changing an inherited epigenetic trait.
It's a powerful demonstration. It shows how nutrition can interact with the genome via the epigenome, with potential implications for human health, maybe influencing risks for things like certain cancers based on early life nutrition.
And it's not just physical traits, is it? What about behavior?
Another huge area. There's growing evidence that early life experiences, especially stress, can induce epigenetic changes in the brain that affect adult behavior and mental health. And again, there's evidence these changes might sometimes be passed down.
Can you tell us about that rat nurturing study? That one always fascinated me.
Yes, the maternal care study. It showed that rat pups raised by mothers who provided high levels of licking and grooming, high maternal nurturing, or high MN, grew up to be less anxious and better able to cope with stress.
Compared to pups with low MN mothers.
Exactly. Pups from low MM mothers were more anxious and had exaggerated stress responses as adults. And the researchers traced this back to epigenetics in the brain, specifically in a gene for the glucocorticoid receptor, GR, which helps regulate the stress response.
What was different about the GR gene?
In the high MN pups, the GR gene promoter in their brain cells had less DNA methylation and more histone acetylation. This open epigenetic state meant they made more GR protein, leading to a better regulated stress response.
And the low MN pups?
They showed the opposite pattern. More methylation and less acetylation at the GR promoter, silencing the gene, resulting in fewer receptors and a heightened stress response.
And this pattern was passed on?
Remarkably, yes. Female offspring of high MN mothers tended to become high MN mothers themselves, and vice versa. It suggests a transmission of behavior through epigenetically regulated gene expression influenced by early life experience.
It just opens up so many questions about nature, nurture, and how they intertwine at this molecular level. So with all this knowledge, where do we go next? What's the future look like?
Well, recognizing the absolutely critical role of the epigenome in health and disease has spurred massive international efforts to map it. Think Human Genome Project, but for the epigenome.
Really? Big coordinated project.
Oh, yes. In the U.S., the NIH Roadmap Epigenomics Project set out to generate reference epigenomes for hundreds of human cell types, looking for patterns linked to diseases like Alzheimer's, autism, you name it.
What are they finding?
Early results are exciting. They're seeing the genetic variants associated with common diseases often fall within regions that have specific epigenetic marks in relevant tissues. It helps connect genetic risk factors to the cell types where they actually function.
And internationally.
There's the International Human Epigenome Consortium,
Yeah.
IUC, coordinating projects worldwide. For example, Germany's DEEP project is looking at metabolic and inflammatory diseases, while Europe's Blueprint project focused on blood cells. They're all aiming to create comprehensive maps.
What about specifically for cancer?
The International Cancer Genome Consortium, ICGC, is doing crucial work there, mapping not just the mutations but also the epigenomes and gene expression profiles for dozens of different cancer types, comparing them directly to normal cells from the same patients.
It sounds like an explosion of data and hopefully understanding.
It really feels like we're entering a new era. These projects are giving us the tools to study health and disease at an unprecedented level of detail, really appreciating how this dynamic epigenetic layer interacts with our fixed genome and our environment.
What an incredible journey we've taken today, exploring this hidden, dynamic world of epigenetics. We've seen how these changes, not in the DNA letters, but in how they're packaged and marked, act like a crucial control panel for our biology.
Yeah, connecting it all, it really emphasizes that while your DNA sequence is fundamental, your epigenome is this constantly interacting layer. It responds to your life, your environment, influencing your development, your health risks, even your behavior. And sometimes, incredibly, passing those influences on.
It just adds such a rich, dynamic layer to how we think about ourselves, health, and disease. So for everyone listening, next time you think about genetics, remember it's not just the sequence. It's also about these epigenetic sticky notes and packaging, this whole regulatory system that determines how your genetic blueprint is actually used. What's the one thing that really stands out to you from our discussion today? Maybe the potential reversibility of these marks for therapy, or how much our lifestyle might actually imprint on our genes. Something to think about. Thank you so much for joining us on this deep dive into the truly fascinating world of epigenetics. Until next time, keep gigging, keep questioning, and keep learning.